Hello everybody, this is Nanduram and we are from Sonu Academy. Today we are going to explain about Organization of Life Introduction part. Children, you have studied in the earlier classes that biology is the science of living organisms. You have also studied the wonderful ways of living of these organisms, the complicated tasks they perform and their role in controlling the environment on the planet. How do they achieve this? Is there something special and unique in the bodies of these living organisms that is making the living organisms different from non-living things? Yes. If you study carefully, both living forms and non-living objects exist together in nature. If you analyze the chemical composition of living organisms, you will notice that they are made of chemicals which are also present in non-living objects. All the living organisms are made up of atoms of carbon, hydrogen, oxygen and nitrogen. Other elements such as sodium, potassium, magnesium, calcium, iron and sulfur are present in small amounts in living organisms. These elements, whether they present in living or non-living forms, but they obey the same rules of physics and chemistry. Example, an iron atom will be the same whether it is present in the human body or in an iron nail. If the atoms of the same chemicals are present in living forms and non-living objects and they obey the same rules of physics and chemistry, then why and how the living organisms are different from non-living forms, and why living organisms can do things which non-living objects cannot do. Similarly, both living and non-living forms are made up of atoms of same elements, but the way they are arranged and organization makes the difference in their behavior. In both living and non-living forms, the organization starts from the level of an atom. In the atom of same or different elements are arranged to form a molecule. However, from here onwards, the organization differs in living and non-living forms. In the case of non-living forms, the organization stops at the level of molecules or proceeds to form crystals or colloids. In biological systems, molecules are organized in a different ways from the non-living forms. Molecules of nucleotides, amino acids and carbohydrates organized to form much larger or micromolecules such as nucleic acids, proteins and polysaccharides respectively from the above simple molecules. These molecules are arranged to form protoplasm. In turn, protoplasm is organized to form a cell. Several cells are organized to form a tissue. Tissues are organized to form an organ, and organs into organ systems, and organ systems into organizations. In complicated organisms, the organ systems, organs and tissues are complicated. Interestingly, the organization in biological system does not stop at the level of organism. Several organisms form a population, several populations form a community, several communities form an ecosystem, and several ecosystems together form a biosphere. It includes all the living organisms on the planet. Hence, in a living system, organization starts at the level of atom and proceeds all the way up to the organisms and above what we explained. Children, having learned the importance of organization and structure in relation to function, let us study in this chapter how the living organisms are organized. We will discuss with the cell, which is the basic structure and functional unit of all the living organisms, and they study how the cells are organized to form higher structures such as tissues in the next coming videos. Points to be remembered in this lesson are: atoms of the chemical elements present in the living things are also present in non-living things objects, and they obey the same rules of physics and chemistry. The atoms of these elements are organized in a complex manner in living organisms when compared to the arrangement in non-living objects. In non-living systems, atoms are organized into molecules and molecules into crystals or colloids. In living organisms, atoms are organized into molecules, molecules to micromolecules such as nucleic acids, proteins and polysaccharides. These molecules are further organized into protoplasm, protoplasm into cells, cells into tissues, tissues into organs, organs into organ systems and then into an organism. Organization continues beyond organisms to form a population, communities, ecosystem and biosphere. In biological systems, structure is related to this function. Cell, the structural and functional unit. In the last lesson, you have learned that cell is the basic unit in structural organization of all living organisms. The fundamental role of the cell in the organisms has come from intensive investigations for the past 300 years by several scientists. Our understanding on the structural and functional organization of cells has increased during this period due to the improvements made on the construction of various types of microscopes and development of a wide range of biochemical and biophysical methods. Let us see what are they. The history of science begins with the description of a thin section called cork by Robert Hooke in 1655. Using a microscope for the first time, Hooke described the cork as being a honeycomb-like structure and called each compartment as a cell which were separated by the wall. As a cork is dead material, cells were empty. Hooke's observation prompted many scientists of that period to observe a variety of material under the microscope. Around the same time, Anton van Leeuwenhoek described the existence of unicellular organisms. Next important discovery was the report on the presence of nucleus in the cell by Robert Brown in the year of 1831. During the same period, Schleiden studied the structure of a large variety of plant tissues under the microscope and observed that all plants have cells separated by a cell wall. In 1838, he proposed that cells are fundamental structural unit in all the plants. Surprisingly, during this period, Schleiden made almost a similar observation using an animal tissue. He observed the presence of cell in the all animals' tissues. These cells have a nucleus but not a cell wall. But for the absence of a cell wall, he observed that the structure of animal was more or less similar to the structure of a plant cell. Both Schleiden and Schleiden discussed their result and proposed cell theory in 1838. It states that all living organisms are made up of cells. In 1855, Rudolf Virchow observed that process of cell division and proposed that cells are formed by pre-existing cells, and this was also added to cell theory. Meanwhile, methods were developed to analyze the chemical composition of a small amount of material. Using these methods, chemical compositions of a wide variety of cells was studied. Surprisingly, it was observed that cells from different types of organisms have the same chemicals in them. They observed the same type of nucleic acids such as DNA and RNA and a large variety of proteins, carbohydrates, ions, etc. This studies indicates that all the cells are made up of same chemicals, and this was also added to cell theory. Studies on the physiology of cells revealed that cells carry physiological functions like they oxidize food materials to derive energy. They secrete the waste material. They proliferate by dividing into two identical cells, and they defend themselves against the attack of foreign organisms, and they try to adjust to the conditions in their surroundings also. These are the same functions carried out by all living organisms. In other words, functions are essential for survival of the organisms and are carried out even at the level of a cell. Function of an organism depends upon the functions carried out by the cell. This concept was also added to the cell theory. Points to be remembered in this lesson are: all the living organisms are made up of cells and their products. All the cells are formed from pre-existing cells. All the cells are made up of similar chemicals and show similar metabolic activities. Functioning of an organism depends upon the functions carried out by the interaction of different cells present in the organisms. As this theory states that all the organisms are made up of cells, we can consider the cell as the basic structural unit of all organisms, since the function of an organism depend upon the functions carried out by its cells. By putting these two statements together, cell is the basic structural and functional unit of all the living organisms. Structure of a cell. The theory states that all the living organisms are made up of cells. We consider the cell as a basic structural unit of all organisms. Since the function of an organism depend on the functions carried out by its cell, we can state that cell is the basic functional unit in all organisms. By putting these two statements together, the cell is the basic structural and functional unit of all living organisms. Having learned that cell is the basic structural unit and functional unit, yes, let us look the structure of a cell. All cells have an outer covering made up of lipids and proteins called plasma. This helps the cell to maintain several molecules inside without getting lost of extracellular environment. Inside the cell, a jelly or a fluid-like substance fills the entire volume of cell. This is called protoplasm. A major chemical compounds of protoplasms are water 70 to 80%, proteins, nucleic acids, amino acids, carbohydrates, lipids and ions. And also a small amount of vitamins are present in protoplasm. The presence of plasma membrane and protoplasm are common to all the cells from bacteria to man or a plant. The bacterial cells differ from the cells of plants, animals, fungi and protists. Cells of these organisms have several intercellular structure known as organelles. Examples: mitochondria, nucleus, lysosomes, endoplasmic reticulum, Golgi complex, plastids and vacuoles are the organelles present in these cells. Each organelle are different. Each organelle has its own membrane. The structure, composition and function of different organelles are different. The major and the most striking difference between bacterial and cell of other organisms is the absence of nucleus. All nucleus is present in bacteria are called prokaryotes, while the algae, fungi, other higher plants and animals are called eukaryotes. The absence of nucleus and other organelles in bacteria representative a primitive conditions, but the bacterial cells carries out the same functions as eukaryotic cells. The structure of a eukaryotic cell. Size and shape of a eukaryotic cell differs considerably. The unicellular eukaryotics have cells in the size range of 1 micrometer to 2 micrometers. The cells of the most of the unicellular organisms are in the range of 95 micrometers to 100 micrometers. A large single cell of multicellular organism is the ostrich egg. It is having 15 to 20 centimeters. The shape of the cell in multicellular organisms are also variously considerable. Most of the cells are spherical or satellite or columnar, means cylindrical. The polygonal or spindle shaped. Cells may also have process of various size extending from its main body. Such long processes are seen in nerve cells. For example, these are the process of 1 meter long in the nerve cells of spinal cord. Of the organelles present in the cell, nucleus is the most prominent one. It can be seen even with an ordinary microscope. It is covered by two membranes which are together called nuclear envelope. Inside the nucleus, there is a diffuse thread-like material called chromatin. It is made up of DNA and proteins. DNA is the genetic material. It has the information required for the synthesis of protein and other vital functions in the cell. This information is present in discrete units called genes. Within the nucleus, a dark staining body called nucleus is present. During cell division, chromatin condenses and forms definite rod-like structures called chromosomes. Nucleus controls and organizes all the functions in a cell. Besides nucleus, there are other organelles in the cytoplasm which perform different functions. These organelles and their functions are listed below. Let us see. Mitochondria. Spherical or rod thread-like organelles. These are called powerhouse of the cell, as they generate the energy required for various functions of the cell. They generate the energy by oxidizing carbohydrates and fatty acids. They have a central matrix covered by two membranes. They contain a small amount of DNA or ribosomes. Lysosomes. These are spherical in shape and are covered by a single membrane. They contain enzymes which degrade carbohydrates, proteins, lipids and nucleic acids. They help in breaking down of complex molecules to simple molecules which can be used by the cell for generating energy or for growth. Lysosomes also participate in the digestion of food material. Under some disease conditions, structure of lysosome is destroyed and this may lead to the death of the cell. Lysosomes are therefore called suicidal bags of the cell. Ribosomes. These are a pear or heart-shaped structures and are not covered by a membrane. They are made up of RNA and proteins. They may be free in cytoplasm or in groups called or attached to the membranes of endoplasmic reticulum. These are involved in protein synthesis. Endoplasmic reticulum. These are tube-like structures present in the cell. They extend all over the cell. Empty space inside the tube is called lumen and is covered by a single layer of membrane. These are two types of endoplasmic reticulum in the cell. Rough endoplasmic reticulum and smooth endoplasmic reticulum to which ribosomes are not attached. Rough endoplasmic reticulum participate in protein synthesis while smooth endoplasmic reticulum is involved in lipid synthesis. Golgi complex. These are also tube-like structures with a central lumen covered by a single layer of membrane. It is involved in the secretion of proteins from the cell. Cytoskeleton. This is a network of fibers present all over the cell. They give mechanical support for the cell and also participate in cell movement. Some of the eukaryotic cells may also have several short hair-like processes called cilia. One or two long hair processes are called flagella. These two structures helps in locomotion. The prokaryotic cell has a tough cell wall. Below the cell wall, there is a plasma membrane. The cell is filled with protoplasm and ribosomes. All the other organelles, include nucleus, are absent in the cell. Long, highly coiled circular DNA is present in the bacterial cell, which serves as a genetic material. Detailed structure of a cell cannot be seen under a light microscope. For this purpose, a special microscope called electron microscope is used to see a structure of a cell. This microscope can show even a very minute structure in the cell as it magnifies the objective by several thousands of a time. Points to be remembered in this lesson are: all the living organisms are made up of cells and cells are formed by the division of pre-existing cells. DNA, RNA, proteins and lipids are the major components of all the cells and the functions performed by the organisms are also performed by the cells. Cell is the basic structural and functional unit of all living organisms. The size and shape of various types of cells are different. Plasma membrane is the outermost layer in all the cells and the cell's interior filled with protoplasm. Prokaryotes have cells without nucleus and other organelles. They have ribosomes. Bacteria are called prokaryotes. Eukaryotic cells have with nucleus and organelles such as mitochondria, lysosomes, Golgi complex, endoplasmic reticulum, plastids and vacuoles. All the fungi, algae and higher plants and animals are eukaryotics. Mitochondria are powerhouses of the cells as they generate energy. Lysosomes are suicidal bags of the cells and participate in intracellular digestion of various substances. Ribosomes are made up of RNA and proteins and are involved in protein synthesis. of group of ribosomes are called polysomes. Rough endoplasmic reticulum has attaches ribosomes and participate in protein synthesis, while smooth endoplasmic reticulum has attached to in lipid synthesis. Structure of a plant cell. Children, in the earlier lesson, you have studied the general structure of eukaryotic cells. Through all the eukaryotic cells have the same basic structure, these are some fundamental difference between animal and plant cells. Let us see what is the difference between animal and plant cells. Plant cells. Cell wall is present in plant cell and absent in animal cell. Vacuoles are present in plant cell and they are present in unicellular animals in animal cell. Plastids are present in plant cell and absent in animal cell. Centrioles are absent in plant cell and are present in animal cell. Cell wall is present outside the plasma membrane of bacteria, algae, fungi and all higher plants. But its structure and chemical composition is different in different groups. Carbohydrates are the major components of the cell wall. The type of carbohydrates present in the cell wall of each of these groups is different. In the bacteria, the cell wall is made up of carbohydrates other than cellulose. In other words, cellulose is absent in the walls of bacteria. In higher plants, cell wall is made up of cellulose, hemicellulose and pectin and lining. Cell walls give mechanical strength to the cell. Without the cell walls, plants cannot support their long and tall body. Moreover, when the plants are watered, cells absorb water and swell. If the cell wall is not present, the swollen cells will burst and die. As the cell wall is tough and hard, it prevents excess swelling of the cell. As a result, the cell becomes slightly stretched and turgid. When there is no water, the cell shrinks and no pressure is exerted on the cell wall. The plant loses the fitness and drops. This is called wilting. Adjacent cells in a plant are connected through a long tube-like structures called plasmodesmata. These tubes are penetrated through the long cell walls. Vacuoles. Vacuoles are fluid-filled bag-like structures surrounded by a membrane. These are present in cytoplasm of a plant cell. In a newly formed plant cell, the vacuoles are small. As the cell becomes old, these vacuoles fuse to form a single large vacuole that fills the entire volume of the cell. In such cells, cytoplasm is in the form of a thin rim around a vacuole and the nucleus is pushed to one side. The membrane surrounding the vacuole is called tonoplast and it allows only a certain substance to pass through it. The fluid in the vacuoles is called vacuolar sap and contain a variety of substances such as carbohydrates, amino acids, proteins, pigments and waste material. Plastids are the organelles which are responsible for the color of the plant cell. Various colors you may observe in plant, especially in flowers and leaves, are due to the presence of plastids in the cells of these parts. Depending upon the structure, color and function, plastids are of three types. Leucoplastids. Leuco means white. These are white color and usually seen in plant parts which are not exposed to sunlight. They are present in the cells of seeds, tubes, rocks and ribosomes. They are of different shapes like oval, rod, spherical and filamentous. They store food material. Chloroplast. These are green in color. They occur in plant parts exposed to sunlight. They contain chlorophyll in the addition to DNA presence in the nucleus and ribosomes. In the presence of cytoplasm, a small amount of DNA and ribosome are also present in the chloroplast. A major function of chloroplast is to trap solar energy and converts this to chemical food energy. As you have studied, chlorophyll plays a major role in photosynthesis. Chromoplast. This contains a fat-soluble pigment which may be red, orange or yellow. They are present in fruits, flowers and are also responsible for their colors. All the three type plasmids have the capacity to change from one to another. For example, young tomatoes are white in color. As they mature, they turn to green and then they turn to red in color. You might have seen green chilies become red and several flowers and fruits changing their color. Centrioles. Cell wall and plastids are present only in plant cells and absent in animal cells. Centrioles are present in animal cells and are also absent in plant cells. In animal cells, two centrioles are present usually on the top of the nucleus. Together they are called centrosome. Centrioles are short, cylindrical structure and participate in cell division. You will learn more about them in the next lesson. Points to be remembered in this lesson. Cell wall plastids and vacuoles are present in plant cells and are absent in animal cells. Animal cells have centrioles and plant cells do not have centrioles. Cell wall is also present in bacteria, but it's structurally and chemically different from that plant cell wall. Cell wall gives mechanical strength to the cell and also helps the cells when they are turgid. Plasmodesmata connects adjacent cells. Vacuoles occupy most of the volume of the cell and are filled with various substances. The membrane surrounding the vacuole is called tonoplast. Leucoplasts store food material while chloroplasts help in preparation of food material by participating photosynthesis. Chromoplasts give colors to flowers and fruits. Chloroplasts have chlorophylls which require for photosynthesis. They also have DNA and ribosomes inside them. Centrioles participate in all cell divisions. Thank you. This is Nanduram and we are from Sonu Academy.